So I'm going to talk about our work in trying to connect the lung and the brain through the process of the Career Development Award. And I have no conflicts of interest for this. I'm going to kind of expand a little bit on the story that Barbara told you about me. So after I joined in 2018 at the pulmonary division of IUCAR, I then submitted my first Career Development Award in June of 2019. Heard back after the music R01 had gotten funded, on which I was not an investigator to avoid any conflict with the K funding. And then heard in October of 2019 that that submission for the K had been reviewed but not scored. And so I submitted a internal Career Development Award, the KL2, in January. And as I'm working to resubmit, I thought it would be a great opportunity to use all of the experience and brain power that's in this room to see if you have any concerns about the resubmission proposal, or if you have any other thoughts from your own experience. So just in case you don't know, I have published in areas of respiratory failure and delirium, biomarkers of ICU delirium, the risk factors associated with both perioperative delirium and ICU delirium, and the management of it, and also authored and co-authored papers on ICU delirium trials and their results. And so that's kind of when they review my application, that's the areas of quote unquote strength that they are looking at. So I just wanted to kind of lay that out there before I start. I also wanted to give a little bit of information as to why this is so important and what the significance and disease burden of delirium is. If you know anyone who's been critically ill, particularly if they're older adults, you know that they can go from being highly functional one day to having significant lung injury due to either sepsis, pneumonia, or another infection, and then be hospitalized and on mechanical ventilation, and their outcomes are much worse compared to a patient who is not on mechanical ventilation and does not develop delirium. And just to reiterate, if you were here for Barbara's talk last month, you'd know this, that delirium is a form of acute brain failure, very similar to another type of organ failure. It's characterized by fluctuating mental status, inattention, altered level of consciousness, disorganized thinking. And here I'm really focusing in on ICU delirium, which is different from some delirium that may be related to an acute stroke, or if the patient is withdrawing from alcohol or drug ingestion. And it's highly prevalent. So up to 80% of mechanically ventilated patients develop delirium. And the US is caring for about 2 million patients a year. And given that the population is aging and we have a culture of demanding aggressive care, this number is expected to increase along with our technological advancements. It is associated with increased mortality, longer hospital stays, fewer days alive and off the ventilator, as well as an increased risk of cognitive impairment, including dementia and related ADRDs. Unfortunately, because the pathophysiology of delirium is not known, or at least not clearly defined, there are no effective treatments, and all of the recent pharmacological trials have been negative for it. And in particular, the patients who have respiratory failure and delirium, we really don't understand the connection and the pathophysiology there, which really limits improving care for these patients. So I'm going to take you back to, if you're not a clinician, take you back a little bit to maybe your biology classes. On the left of the screen, you're going to see a normal alveolus, which is the grape sac at the end of a lung unit in your lungs that helps to take air that you breathe in across the bloodstream. Whereas on the right side of the screen, you're going to see the current model for acute lung injury. And that model suggests that due to whether it's an infection or you've been in a motor vehicle accident, or you have another organ failure such as pancreatitis, you have cells in your body called macrophages that are staying in the lung looking for signs of invaders. And when they see that, when they see a threat, they start to release inflammatory molecules that we call interleukins or cytokines. And they cause other cells, such as the neutrophils, to get excited. And when they get excited, they release molecules that are very similar to hydrogen peroxide. So these are oxidative species that cause oxygen radical toxicity, and they cause more injury to the lung circuit. And when they do that, there is even more inflammation, and that cycle perpetuates. This actually has great parallels to what we think is happening in delirium, where you have those same inflammatory cytokines interacting with the blood-brain barrier and causing this, the brain's corollary cells to react, get excited, cause more local tissue damage. And if it's a short-term event, you may get delirium, whereas if you get prolonged inflammation, it can lead to dementia. So there's a problem in this theory, and the problem is that patients who not all patients who have respiratory failure develop delirium. So we think that there might be a balance between anti-inflammatory and pro-inflammatory cytokines, and it's the patients who are having brain failure and respiratory failure that have more of the inflammatory cytokines. What that does is causes oxidative damage, and we already know that that oxidative damage is hurting the DNA. And when you have DNA damage, you then have protein damage. And proteins are the mechanisms by which the cells actually perform their functions. So if you break down cell activities, you then develop organ failure. So that is that's the hypothesis here, which has been backed up in mice models. So my original submission, I'm going to kind of show you the original submission concerns, and then I'll show you what our planned submission is, and welcome your feedback. So the original submission was very ambitious. It was a music trial, which was mechanistic. And this is before our DDM R01 got funded. So it was using the FOA for a K23 with a clinical trial, with a 2-arm randomization using the preliminary data we already had supporting slow tempo music versus an attention control. And the mechanistic component was to look at the effect of the slow tempo music on reactive oxygen species related damage, which was DNA damage. And the career development plan component laid out my career goals to be a clinical translational researcher. The main critiques from the initial submission, number one was there's a concern about the scientific premise of the proposed music clinical trial. The reviewer was not convinced that the biological pathway of music leading to a reduction in inflammatory cytokines would end up leading to reduced DNA damage, and then downstream would actually improve outcomes. And they wanted a more compelling conceptual model, better preclinical data, and an understanding of how this would fit into larger work. And if you look at the original aims, they were very much tied into music. So what's the effect of the music on DNA damage? What's the effect of music on plasma protein expression as a downstream indicator of DNA damage? What's the effect on the cytokines causing reactive oxygen species? And then how does music relate to clinical outcomes? And if you look at a conceptual model, our conceptual model was, well, these patients get respiratory failure. The music is going to help reduce inflammation. If it reduces inflammation, it's going to reduce DNA damage, and so on. And the yellow boxes that you see are the different aims that we had measured in the study. And our plan was we would take 96 patients in, we would randomize them, and we would collect blood on day 1, day 4, day 7, before and during the interventions, and basically plot out their DNA damage against their delirium outcomes. So now for the resubmission. So we have really reworked the design. And this is the 30,000-foot view. So we're proposing to change the study design to a secondary analysis of data that's already been collected, so blood samples that were already collected, as well as an observational component where we prospectively collect them. Connect the aims, remove music entirely, and connect the aims to DNA damage and delirium. Emphasize the infrastructure that we have because Babar Khan and the rest of the group have developed a huge biorepository of samples from ICU patients, and emphasize that there's already 2 ongoing clinical trials in the ICU that can help get these samples for us, and emphasize the pilot data that we now have. So the resubmission response, the aim now changes to evaluate whether reactive oxygen species mediated DNA damage is going to be independently associated with delirium duration and severity. And this graph, if you like to look at it visually, basically just plots out the hypothesis that as we see more DNA damage, we're going to see increased delirium duration, we're going to see increased delirium severity. Similarly, if we see more DNA damage, we're going to see more length of mechanical ventilation, longer stay in the ICU, as well as fewer days on the vent. And here I'm showing secondary specific aim 2, where there is now another component which looks at biomarkers of acute lung injury separate from reactive oxygen species, so that we're not putting all our eggs into the ROS-mediated DNA damage bucket. So as these biomarkers of lung injury increase, delirium burden and duration of mechanical ventilation would increase. So this is a revised conceptual model where you have acute lung injury, they have more biomarkers of endothelium injury, they have increased pro-inflammatory cytokines, more ROS-mediated DNA damage, and delirium duration, severity, mechanical ventilation, and length of ICU stay outcomes. And the design is patients who have respiratory failure. We have a large number of samples already collected from the PMD trial that was done here at Eskenazi as well as Methodist and University. And we are already prospectively collecting samples from patients who are put on mechanical ventilation due to respiratory failure. So these are samples we collect at enrollment to the study, which is as close to mechanical ventilation time as possible, and then day 7. And we'd be able to pool the 2 samples together to increase the power. The inclusion criteria would be our usual: admitted to the ICU, adult patients, diagnosed with respiratory failure, exclude the patients we normally do in our delirium trials to avoid confounders. And the next slide I'm going to show you is the preliminary data that we have been able to find. So since over the past couple of months, we have collected blood samples from patients admitted to the ICU on the ventilator with respiratory failure. And what I'm showing you here is the amount of DNA damage these patients have. So PBS is a control. So this is just patients who there's no patient, so it's basically just plasma from normal patients. And then number 74, number 114, and number 43 are different subjects. And these are the patients who had delirium and respiratory failure. And you can see that there's about 30% DNA damage in patients without respiratory failure or mechanical ventilation. And it's somewhere between, it ranges as high as about 55% in patients who do have respiratory failure and delirium. And these differences are significant. We are working on increasing the sample size for this pilot. We've just had some issues with some assays, but this shows that we can find DNA damage in these patients and that we can also do the assay. This is graphically a picture of what the DNA damage looks like. So at the tip, you'll see this kind of a bright dot. That is the nucleus of the cell, whereas that long tail that you see is due to DNA damage. So the higher the DNA damage, the more of a tail we're going to find. One final point that I want to show you, and then I'd like to stop and ask you for your input. The second concern was that the training plan is too broad and it risks being shallow because I had laid out that I would obtain training in lab skills, patient enrollment, bioinformatics, more of computational analysis with that, cytokines and proteomics expertise, and clinical trials experience, which the reviewer felt was unrealistic, that I would be able to fit all that into a 5-year K. So the revised candidate objective is to be a nationally recognized and independently funded translational clinical researcher connecting the respiratory failure and delirium pathophysiology. Here on your left side, you're going to see the original goals, and then in the blue box, you're going to see the revised goals. And so the revised goals really focus on biomarker development. So developing skills and training in translational science, conducting biomarker selection, analysis, and identification, and then training in models of oxidative DNA damage, as well as DNA damage in patients who have delirium and respiratory failure, aligning with areas of strength from my publications. And then finally, the third goal, training in leadership and transition to become an independent investigator. We have also done a realignment of the mentoring panel to kind of focus the reviewers on the strengths of the team. So Barbara Khan has expertise in delirium biomarker work and as a translational researcher as well. Malaz would be adding in expertise on delirium and transitioning to an independent investigator. Sugao provides the biostatistical expertise in biomarker computation. Roberto Machado, who is the chief of the Division of Pulmonary Critical Care and also on the mentoring panel, he has expertise in molecular models in sickle cell disease and pulmonary hypertension. So he would be able to connect the lung disease components. And then Michaela Aldred, also from the Pulmonary Critical Care Division, her expertise are in lung injury, oxidative DNA damage, as well as repair of injury mechanisms. So putting that all together would focus, we think, would focus down the aims. So I'm just one slide away from stopping here. I'd love to get your response to the reviewers on the research plan and your response to the revision of the candidate training plan if you feel that we're too narrow, too broad, still too ambitious, or any other feedback you might have along the way based on all of your experience. I will just remind you, the registration just opened for the American Delirium Society meeting that's going to be here in town in June. So I hope you'll take the time to register. If for some reason you don't want to speak up here, please email me if you have any thoughts, or you can also tweet. Thanks very much. We still have 10 minutes, so I'm willing to stand and wait.
I mean, so it seems concordant with, they thought we were premature for a music therapy trial. And so you're, because they, as you had acknowledged that the mechanisms aren't well understood, so you're trying to do that. One very specific question, since you've realigned your training plan now, you have a terrific group of mentors. They're usually going to want to see some specific courses as well. You don't have to mention what they are, but are courses available here at IU that are aligned with your new training plan?
That's a great question. There are some amazing courses here, thanks to the School of Medicine and Purdue. So I've actually put it's kind of hard to fit on one slide, but I basically have courses that they thought were too broad initially, like there's too many courses. And so I have, and they also didn't like that some of the courses were somewhere else remotely online. So I've really narrowed it down to give me depth of expertise in translational science and biomarker development and biomarker analysis. So that is fortunately available here. The one area that we're not as strong in that they wanted to see is grant writing courses. As you know, because of the SITE program, I already took one, and there's not a lot of other strong courses locally. There may be one down in Bloomington, so that's what I ended up adding on.
The CTSI has developed a grant writing course. I mean, I'm not involved in it, but they have it for people doing K awards, and they did it last year. They'll do it up. So just put that one down.
Sure. Now, do you know if is that a workshop or a course?
It's like 8 sessions or something.
Okay.
I don't think it has a G500 associated with it, but you could get Lane Coffey. He can get you the description, and I would just write that into the grant.
Great. Thank you.
So along that note, there are individual departments and schools also have grants courses. So we offer in the School of Public Health, there's a grant writing course for students in that. And there's a School of Science has a grant writing course that they offer to their PhD students. So if you can't find one through CTSI, then you may be able to go to an individual school and to add that to your plan.
Thank you. Yes.
So I don't know, do you have a slide that shows your revised research aims along with your revised career goals?
So this slide shows the revised goals.
Okay.
So the one in the blue box is the training goals that are revised.
Okay. And then your revised specific aims.
Right. So these are the revised aims.
Okay.
And then I have the original ones on a separate slide. Would you like to see the original ones?
No, I was just, so I think sometimes, I mean, if you've made significant changes, obviously on the project piece, and I would just sort of, I think it's really helpful when you can sort of align the fact that completing the aims will actually help support the career goals.
Oh, sure. Right. Yeah, I agree. And the write-up in the career development plan is trying to do that, but I'll make sure that it's a bit more clear, even as I'm going through the research plan. Is that answer?
Yeah.
Okay.
I think sometimes when you can sort of show how they need to get
Right.
the research aims are meant to help you.
Right. Exactly. Yeah.
Since I've recently gotten feedback on a K23, one of the things that they seemed to focus on a lot was being very product oriented and also benchmarks. So making that in that table more clear, and some of the other things that are more routine, you could probably take out and just keep in a written form in terms of like mentorship meetings, frequency and stuff like that. And even if you can say like which mentors are going to be co-authors on which papers.
That's great. Thank you. I've tried to put it as much as I can into this table for the presentation, but then the rest is in write-up. But one thing I haven't put in here is who's the co-mentors on those papers, so I can certainly add that in. Okay. Well, thank you very much.